Hello to all. Today we are going to discuss about some MCQs which are based on the molecular biology. The first question is that amino acid sequence in protein synthesis is decided by the sequence of. So always remember that the sequence of the amino acid in a protein chain is decided by the sequence of the codons found on the mRNA. Say for if this is the mRNA and this is the codon and this is the charged tRNA, and charged tRNA is containing what? The amino acid, and this is the anticodon site. The anticodon will be just complementary to the codon. So always remember the sequence of the amino acid in a protein chain will be decided by the sequence of the codons found on the mRNA. So what will be the answer? That amino acid sequence in protein synthesis is decided by the sequence of what? tRNA, mRNA, cDNA or rRNA. So always remember that the answer will be B, that is mRNA. So the question number 1 is having the answer B. Now polysome is formed by, very first you must know that what is a polysome. So polysome is also called as polyribosome. Polysome is also called as polyribosome. And how a polyribosome is formed? So this is the mRNA, and attached with this mRNA are many ribosomal units. Okay, these are the ribosomal units, and this is what? The mRNA. So this is a polyribosome. So polyribosome means, poly means many, ribo means polysome, means many ribosomes are attached to each other with the mRNA. So polysome is formed by a ribosome with several subunits, a ribosome attached to each other in linear arrangement, several ribosomes attached to a single mRNA, or many ribosomes attached to strand of the ER. So what will be the answer? That is several ribosomes attached to a single mRNA. This is what? The mRNA, and these are the ribosomal units. Okay, so the answer C is correct for the question number 2, that several ribosomes are attached to a single mRNA. Question number 2 is having the answer C, right? Now the next question is, which of the following bond is not related to the nucleic acid? Now when we study the nucleic acid, we study about the different bonds, and the bonds which are found in the major bonds which are found in the nucleic acids are, first one is the hydrogen bond, the second one are the glycosidic bond, and third one are the ester bonds. Okay, now the nitrogen bases are attached to each other with the hydrogen bond, the sugar and the nitrogen bases are attached to each other by the glycosidic bond, and the phosphate and the sugar are attached together with the help of the ester bond. Now see here, which of the following bond is not related to the nucleic acid? Hydrogen bond, it is there, ester bond, glycosidic bond present, but peptide bond is not there. Why? Because we know very well that peptide bond is never found in the nucleic acid. Peptide bond is found in the amino acids and it forms the protein chain. So peptide bond will be associated with the protein, not with the nucleic acid. So the answer is the D. Question number 3 is having the answer D. Now removal of introns and joining the exons in a defined order in a transcription unit is called as. Now what you have to see here, that in question number 4, the question is for the introns and the exons. We know very well that there is a RNA known as the hnRNA. hnRNA means heterogeneous RNA, and it consists of two types of the sequences, right? One are known as the exons and the another one are known as the introns. Exons, introns, then again exons, like this type. So a heterogeneous RNA is having the two type of the sequences, exons and introns. Now these exons are expressed sequences. Okay, these are the expressed sequences and these are unexpressed sequences. Now, if we want to pass the genetic information from one exon to another exon, it is not possible. Why? Because the unexpressed sequences known as the introns are present in between the two exons. So the removal of the introns is called as the RNA splicing. Removal of the introns is called as the RNA splicing. So the question was removal of introns and joining the exons in a defined order in a transcription unit is called as, so it is splicing. Question number 4 is having the answer B. Okay. Now, which one of the following makes use of RNA as a template to synthesize the DNA? Means the question is very simple. The question is that which enzyme is responsible for the formation of DNA from RNA? Means say for we know very well that from DNA we form RNA. But sometimes this RNA can act as template. And instead of forming DNA forms RNA, but sometimes the RNA may form the DNA. In this case, the RNA acts as template, and this happens in the presence of an enzyme known as the reverse transcriptase, and the entire process is called as reverse transcription. So what is reverse transcription? Which occurs in the presence of the reverse transcriptase enzyme. That in this process RNA acts as template and forms the DNA, right? So which one of the following makes use of RNA as a template to synthesize DNA? DNA dependent RNA polymerase, reverse transcriptase, or RNA polymerase? So the correct answer is the C, reverse transcriptase. Question number 5 is having the answer C. So these questions were based on the molecular biology. Hope so you have liked this. Thanks a lot for watching. If you want to take the screenshot of this video, you can take.